Good afternoon, and thank you for joining. So my name is Linda Kouvlar, and I'm happy to be your host for today's webinar, during which we will explore together how OncoDNA and MGI are helping to shape the future of precision medicine in oncology. And today we will also have the honor to have Alejandra Maria Perez-Sast, who is Services Direction Manager at Sistemas Genómicos, who will share her experience. So, as you know, NGS has opened the door to powerful diagnostic and treatment opportunities in oncology, but putting this technology into routine clinical practice is not always straightforward, and mainly because of technology availability, sample quality, and also the complexity of the data analysis. And that's exactly why the collaboration between OncoDNA and MGI is so relevant. Not only because this partnership enabled more affordable access to NGS technology, thanks to, and also with all the benefit provided to the DNBSEQ technology, but also by providing better insights with the OncoDEEP and OncoKDM solution from OncoDNA. So let me present you the speakers of today. So Konrad Iken is Chief Product Officer at OncoDNA and spans a range of roles in data analysis companies specialized in NGS. And he will open the webinar by presenting you the OncoDEEP and OncoKDM solution. Philips Oleskovs is Business Development Manager. He drives IVD solution project at MGI, and he will present the DNBSEQ technology and how MGI sequencer enable to obtain more precise information. And our last speaker will be Alejandra Maria Perez-Sast, Services Direction Manager at Sistemas Genómicos, now part of Eurofins Group, and she will present data obtained from clinical samples. So at the end, a Q&A session will be held, and I invite you to use this dedicated channel to share your question. So now let's start this webinar. Konrad, the floor is yours.
Okay. Thank you, Linda. So it's a pleasure to introduce OncoDNA and our solutions to the audience. So today we are going to focus on the different solutions developed by OncoDNA and how we collaborate together with MGI. So the main focus will be on our kit solutions, which we sell to laboratories all over the world. But first, let me give you an introduction on the history of OncoDNA. So actually, OncoDNA started as a company that provided reports for oncologists, meaning that the report needs to be very clear, concise, and the actionable biomarkers needed to be available quickly so that an oncologist, when discussing with a patient, could make a good judgment on the next steps for this patient. So this is why we have a report which shows the clinical trials, which biomarkers we see very clearly on a first page. And then, of course, if an oncologist would like to go and dig into the details, they can go down to the 17 or 20-page long report to look into all the details of the variants we have found. Or they can look into the OncoKDM platform, which is a web-based platform where all the information is also available through clicking through the platform, which is very convenient for an oncologist to quickly go to the direct section to find the information. And like this, we have been able to serve many institutions in many different countries, and OncoDNA is really, even though we are a Belgian company, not at all focused to Belgium, but we are working and collaborating with hospitals and institutions and pharma companies all over the world. So a little bit of history on how we have been able to make these reports and these services. It started in 2013. And of course, the first thing you need to do when you would like to build a solution or a service like the OncoDEEP solution or the OncoKDM solution, you need to have the knowledge available. So this is why the team started by creating a large database, the OncoKDO database, where we gather all the information which is available about specific variants linked to treatments or specific biomarkers linked to treatments, clinical trials, scientific information. So even today, after 12 years, this database is still the core of OncoDNA, and each day we have algorithms combined with our scientific team who will go through all the new information available on the oncological testing. So once this database was built, we started to develop different tests which we ran in Belgium in our laboratory. So based on what we knew about the biomarkers that needed to be covered, which variants are important, which genes needed to be in the panel, we designed our first OncoDEEP solution, which was specifically made for solid tumors. This panel has been optimized to be used in our lab in Belgium, and then we developed the BioIT and the web interface to distribute the BioIT and the reports to the oncologist. So we've been working for a long time in this manner, but then actually, as of the 7th iteration of OncoDEEP, we made a change in our solution. So there we moved to a twist-based, capture-based solution for the OncoDEEP V7. And this also allowed us to work together with other sequencing machines in the field. And this test is now as well compatible with the MGI sequencers which are available in the field. So now we have basically two business lines which coexist next to each other, or we ship samples from oncologists to Belgium and we run the test in our lab. The BioIT is processed in a cloud system, and then the report is visualized through the OncoKDM system to the oncologist. Or we can send you the reagents that each laboratory can use the test in their own laboratory, on their own equipment, and then the FASTQ files will be uploaded into the same cloud-based system. The same BioIT solution will be used to generate the results, and OncoKDM will be used to visualize the results for the experts in the lab. Of course, this shift in business model and approach needed also changes to our software solution, because now we had clinical geneticians, we had experts in molecular biology looking into our system, and they wanted to go into the details instead of just finding the clinically relevant or actionable mutations for a specific patient. So this is why the OncoKDM system has seen a dramatic evolution in the last years, where we now also have a lot of functionalities which are very interesting for laboratories. For instance, a user can look into the biological impact. He will see all the details of these specific variants with the variant frequency, the naming, the impact, and then a lot of information on the specific variant with the links to the PubMed IDs. Another example is, for instance, the CMV calling, where we don't just say there is a CNV, but we give you an exact visualization of the different exons and what is the copy number of the complete region. So all of these changes have improved the platform and made it more fit for the experts in the lab to use the OncoKDM platform. Of course, the oncologist can also still log into the system and very clearly see the information immediately without going into the details. What's also very important, as OncoDNA, before we make a decision based on a variant, it needs to pass certain thresholds. This is again used when we make a report for an oncologist. Everything which is below a certain threshold is not taken into account to make the call. If you are working in a laboratory, of course, you would like to see all of those details of those variants. So you have a specific tab which is called All Variants, and there you see all the variants, even the ones that are below the predefined clinical thresholds set by OncoDNA. And then you can decide to include this specific variant, even though if it does not meet the predefined thresholds, to include it into the report. For laboratories who want to go further, you see on the right-hand top side, there is the possibility to download all the files which are being generated by the BioIT pipeline, and you can investigate even further based on the raw data of the BAM file, the VCF file, and you can look into the details of the Fusion file. So all of these changes we have done during the last 2 years when we moved towards the kit business really makes us believe that we say we put our knowledge in your lab. So all the expertise OncoDNA has gained in how we analyze samples in the lab, how we design a gene panel, how we design a BioIT pipeline, and how we do the reporting is now basically put into a little blue box and can be shipped over to the lab. You do the wet lab work yourself in the laboratory, and the bio-IT analysis and the interpretation is based on the same system as what OncoDNA already uses for more than 10 years in our centralized setting. So a little bit more on the OncoDEEP kits. As said, we focus, of course, on oncology samples, and each OncoDNA kit consists of reagents, a bio-IT pipeline, and an interpretation system. The reagents we deliver are meant to start from extracted DNA or from FFPE blocks or from blood samples. And you have all the reagents to do the library preparation, the target enrichment, and then you use those captures on your own sequencing platform. Depending on your throughput, you can have access to a bigger sequencer or a smaller sequencer. Once the FASTQ files are generated, they are uploaded into the cloud-based system of OncoDNA, and after 48 hours, you get a report with all the relevant information for this specific patient. A little bit more information on the bio-IT pipeline. So it's we do all the checks, the QC checks, alignment, variant calling, and then once we have defined the variants that meet our quality thresholds, they are being referenced with our OncoKDO system, and this will be used to generate a report and validated by our clinical team internally before it's being released to the end user. A few of the examples of the biomarkers, the complex biomarkers we target are listed below. So we can detect HRD, MSI, TMB, CMVs, LOH with our OncoDEEP solution. And also an important topic is the alpha list, because sometimes it's also very important to know what is not available on specific, very important positions in the genome. So our alpha list, we detect all the variants, we visualize all the variants, even if they are below our thresholds, to make sure if there is a resistance position or a very important position, that each user can specifically look at this position, that we don't miss anything. This is an example of how, as OncoDNA, we have tailored our platform to be really used in an oncological setting, and this is also why we only focus on oncology samples, and we do not do hereditary testing. And again, as said, the core, which is not really exposed to our end users, is the OncoKDO, where we gather all the knowledge we have as OncoDNA, and through which we will provide this information to the end user. So what do we have currently available as OncoDNA? We have our flagship product, which is the OncoDEEP solution. We already tested more than 8,000 samples in the decentralized solution with the OncoDEEP. It's a panel which consists of 638 genes on the DNA level, and we also have an RNA component which has 22 genes included. The combination of the DNA and the RNA is a very cost-effective solution to analyze the most relevant biomarkers in an oncological setting. The DNA also has some translocations on the DNA level, so we can detect ALK, ROS, RET, and MET exon 14 skipping on the DNA based. And then, if this is not sufficient, you can continue with the add-on of the smaller RNA panel to expand the solution. One thing which is not included on the slide, but which is very important as well, actually we have gotten a lot of feedback that the third promoter, which is also very important, is a very difficult target to detect on an NGS based. So this is why we've put extra effort in the design of the panel to make sure that we also can detect very well the third promoter. And we are one of the only panels which detects this third promoter in such a good way. Here you see a little bit more detailed information on what we expect of the sample input. So we need to have at least 30 nanogram input. We have certain quality parameters we expect before we claim that the result can be given. So the DV900 or the DV200 for the RNA is very important. And then if we have these inputs, these good quality samples, we can detect the variants that we need to detect at a coverage of 350x. Because of the good uniformity of the twist solution, 350x is sufficient to detect all the variants we need to find. If you look at the throughput, so we on the G400, the small flow cell, we can run 16 samples at once. On the larger flow cell, we can run 96 samples. On the G99, which is a smaller sequencer, we are currently limited to 6 samples that we can analyze on this sequencer. Each OncoDNA kit consists of 4 captures of 8, meaning you can run 32 samples with one OncoDEEP kit. Another solution which is already currently available is the OncoSelect. It's a liquid biopsy panel. So of course, for a liquid biopsy panel, we need to sequence way deeper than with a solid biopsy. So here we aim at a coverage of 20,000x, which means that we have less genes in the panel. So it's a panel consisting of 74 genes, where we can also detect the SNPs, indels, certain fusion genes. We have put the same effort on the third promoter, and we can detect the HRR genes. A little bit more information on the OncoSelect library specifications. So again, it's based on the twist bioscience chemistry. It can be sequenced on a 2x150 base pair flow cell on the MGI sequencers, and we need to have UMIs into the panel. We start with 20 nanogram of blood, and we need at least 120 million paired end reads to do the sequencing and to be able to get the quality criteria we need and to be able to correctly detect the variants and the alterations in this specific sample. As said in the beginning, we run these solutions also in our own lab. So we have data available, which you can find in a white paper, which is downloadable through the website of OncoDNA, where we give you the performance characteristics of over more than 2,000 samples which have been analyzed with the OncoDEEP solution. It can give you some insights on what we use as, or what we see as quality needed for the samples before we start the analysis. So this is what we have available and what is being used in Europe and in different parts of the world. But we also have a philosophy for our future product pipeline. So as you saw in the beginning of the presentation, we are looking to give the best test and the most relevant test for oncologists and for laboratories serving oncologists. So this is why we are developing our product portfolio, and we want to use the same philosophy for all our tests. So we always want to have one flow starting with the same reagents, potential automation in the lab, sequencing, then the analysis through the OncoDNA cloud-based solution, and the clinical interpretation based on the information which is available in OncoKDO. So for the solid biopsies, we have the OncoDEEP Kit, as mentioned before, and we are now developing also a panel based on the whole exome sequencing, where you can run the complete whole exome sequencing and do the same bio-IT pipeline and use the same systems as the OncoDEEP solution. I mentioned the RNA panel, the 22-gene RNA panel. Now we are also launching a more than 400-gene large RNA panel, which is demanded by a lot of our customers. In the liquid biopsy, we have the OncoSelect kit, which I explained before, and we are also looking into the possibility to launch a 400-gene liquid biopsy panel as well, which will be dedicated for treatment selection. And then another area where we are going to focus on is in the liquid biopsy solutions, the monitoring, where we will have a tumor-informed solution, what we call the OncoFollow. It means that we will first do an OncoDEEP or an OncoDEEP whole exome sequencing for a patient, define which variants we would like to follow, make a specific panel for each specific patient, and then we can follow up with this personalized panel. We're also looking into the possibilities with a university partner in Europe to see if we can make a tumor-agnostic MRD solution, where we will also do the follow-up of specific persons, but where we do not need to know the details of the tumor before. And then all of this all of this, the data which comes out of these solutions and the information will be gathered into our OncoCDI, which you see in the middle, the data intelligence tool of OncoDNA, which will allow users to explore the data in their own lab and compare it with other statistics we can provide them with. Of course, all of these solutions, they are built around sequencing. So we can only give high quality results if we have high quality sequencing done in the lab. And this is the collaboration we have now with MGI. So we can provide the support to users who have MGI sequencers. And now I would like to hand the speech to Philips, who can give more information on the availabilities of MGI in the market.
Thank you, Konrad. So at MGI, we are striving to lead life science innovation and make NGS-based diagnosis for as many patients as possible. And while our core technology, core business is sequencing, we're also providing automation solutions. So we have these two main segments, and automation solutions are supplementing our sequencing portfolio. So in the end, we are able to provide whole integrated solutions from a very low to actually highest in the world of throughput. And in the essence or in the core of MGI is our technology. It's called DNBSEQ. And here I would like to guide you through how it works. So as usual, when we are doing library prep, we're getting fragments of double-stranded DNA library. And in our technology, it is circularized. And from this circle, using linear amplification, we are making a very long stretch of DNA that contains copies of the insert, multiple hundreds of copies. And this long stretch of DNA in the end is condensed to form DNA nanoball or DNB. And DNB is basically the core of the technology that we're using that has some great advantages as well. So after the DNB is made, it can be loaded on a flow cell and sequencing is happening. So apart from, for example, avoiding index hopping, this method also allows for increased accuracy of the results, and it also decreases duplicate rates in the sequencing data. And this is also achieved due to uniqueness of DNB technology that doesn't rely on the amplification in the chip, and the amplification which leads to DNB making is actually very precise. And these advantages are specifically important for clinical studies or diagnosis because, as you know, in clinical real world settings, samples are not always perfect. And this increase in quality and accuracy of data is very important, especially when working, for example, with FFPE samples. So in this collaboration that we have with OncoDNA and also supported by Sistemas Genómicos, we used G400 machine from MGI. So it's a middle throughput sequencer. And samples were collected and prepared at Sistemas Genómicos using OncoDEEP panel, then sequenced on MGI machine and analyzed on the OncoKDM platform. And apart from G400, we are also having developments on G99, which is a smaller throughput, very quick machine where we already have very promising results in the proof of concept. And as well, we are developing a script for SP100 automation. This is a smaller version of our automation machines that in the end will allow to have a seamless workflow, fully automated, that can also be introduced in clinics as it can be fully IVD certified. And so this is the theory, and I would like to hand over to Alejandra, who will tell more about real-world application of this pipeline.
So thank you very much, Philip. Thank you very much for the invitation to participate in this seminar and for the opportunity to share with all of you our experience here in Sistemas Genómicos in the implementation of the service in our company that includes also the workflow validation of MGI technology. Well, if I may, I would like to make a short introduction about our company. Sistemas Genómicos is an international biotech company located in Valencia, in Spain, with more than 25 years of experience in genetic diagnosis. And historically, we are a service providers company, but that is with solutions that cover the entire human life cycle. We are speaking about preconception, human reproduction, cytogenetics and prenatal studies, and also postnatal studies at germline and somatic level. Well, we are also a quite multidisciplinary team. That means that we are not only geneticians, but also bioinformaticians, cloud experts, application developers, etc. And all this multidisciplinarity gives us the capability to packetize all the developments that we need to be used in-house for our services and offer end-to-end solutions that can be implemented in laboratories or centers with technology transfer or directly implementing our technology with more than 25 kits, all of them certified with CIVD. Well, as a laboratory offering a highly specialized service in genetic diagnosis in Sistemas Genómicos, we are quite well aware about all the challenges and requirements that a somatic sample analysis service must meet. The service will provide clinicians with all necessary information to make the most appropriate decision tailored to the patient tumor profile. And for these reasons, it is necessary to provide a series of conditions that this is quite important to obtain this information as soon as possible. The first one will be an immediate clinical relevant results that directly support therapeutic decision. That means target therapies, immunotherapy, clinical trials, as well as a complete interpretation of the results aligned with up-to-date clinical guidelines, the NCCN, ESMO, ASCO, etc. Well, second point, it is necessary to use comprehensive genetic solutions with comprehensive panels that include all key relevant genes related to the different tumor types, and also the ability to detect multiple alterations types, simple nucleotide variations, indels, copy number variations, fusions, etc. That with one single solution will have all the information that is necessary for the interpretation of the information related to the therapeutic decision. Then, next point, we should always keep in mind that behind this kind of services is always a patient that is waiting for a treatment, and therefore the fast turnaround time is so necessary in this time of services. And we need to work with time delivery results that should be ideally between 10 and 14 days, and also offer a clear and conscious reports that do not require further interpretation from the oncologist. That it should be very easy to identify which is the treatment that is necessary to select for a patient according to the tumor profile. Also, include complementary immuno-oncology profile with these genomic signatures, these biomarkers such as HGRD, TMB, MSI, that Conrad explained that before, that this is included with when we are implementing the OncoDEEP. And finally, but one of the most important points, even if we will have so much information at OncoDNA or the tertiary analysis that we can obtain with a solution is so comprehensive it is always mandatory to have a strong technical and clinical support behind all these kind of reports that offer an expert consultation especially for complex case of interpretation. Well, if we look at the entire genetic diagnosis workflow, and especially considering the new European regulation for in vitro medical devices, the whole process should fall within this new framework. But in other words, what that means is that all equipment, reagent, and software must be CIVD certified. Taking into account all the necessary requirements that I already explained, as well as the regulatory framework within which we must operate, the only solutions that meet all the requirements was OncoDNA currently. However, at the time that we are evaluating the different options, the methodology had not yet validated on MGI chemistry, and therefore it was necessary to carry out a validation using the DNBSEQ-G400, that at the moment was the only open sequencing platform with CIVD certification. Actually, a few minutes before, Philips explained that it's also possible to run in the G99, but at the moment that we start with this validation, the only one that have the CIVD on the stage, it was the G400, and therefore it was the one that was used for this verification. Well, to carry out this verification of the technology used in the DNBSEQ-G400, it was to use clinical samples previously analyzed with Illumina technology. The idea was to take exactly the same libraries that it was run before with this technology, and also to use the same bioinformatics secondary and tertiary analysis. Then the only variable that we will introduce, it was the sequencing, and so it was more easier to perform this comparison between the impact it could introduce the new chemistry during the sequencing and the impact in the results that we are obtaining. But it was to introduce the samples with no single nucleotide variants, copy number variations, fusions, and genomic signatures previously detected with OncoDEEP Kit combined with Illumina sequencing, to cover all the different types of variants of information that we will obtain when we are performing this kind of tests in our facilities to offer a complete and comprehensive solution that the clinicals are looking for. The reference standard were also used, 6 from Horizon for variants and fusions, and 3 from Seracare from tumor mutational burden for the calculation of TMB. And it was also tested different DNA inputs using 6 nanograms, 30 nanograms, and 100 nanograms. If you if you use this kind of you are implemented or using this kind of services of methodologies, you will know that the material, the star material that we are using for somatic studies probably is one of the most handicaps, not only for DNA, but especially for RNA. And then the amount of quality sometimes is not the best one. Therefore, it was necessary to establish this range of different DNA total amount that we can use to perform the procedure. So we will very short which one, where is the limit when we perform or implement the technology in the laboratory. At the end, it was a total of 90 samples, 54 clinical and 36 reference samples that were sequenced to evaluate the performance of the MGI chemistry. Well, the results after the sequencing, it was we have implemented the technology in MGI. Then you can imagine that the results were very, very nice. Actually, we were looking for 3 different analyses. The first one is the technical one for to evaluate the quality of the data that we are obtaining in terms of electrons on target, PCR duplicates, reads, coverage, all these kind of parameters, and we obtain a very nice high quality data of metrics, especially for the total amount, the input of DNA between 30 and 100 nanograms. It's true that with 5 nanograms, with 6 nanograms, the results were so nice, especially when we are speaking about coverage and PCR duplicates. Therefore, they have too much duplicates, and then the coverage it was impacted, and we saw less coverage than with 30 and 100 nanograms. Then most of the variants on fusion were detected in clinical and control samples, and those that were not identified were only due to a low amount of the starting DNA. It was done with 5 nanograms, and it was not a workflow issue. And also, and it is also important to mention that the ones that it was not before and that it was not identified with 5 nanograms was exactly the same problem with Illumina. With 5 nanograms, it was not possible to identify it also with Illumina chemistry. Then, next point, the calculation of the genomic signatures, the TMB, MSI, and HRD. For HRD and MSI, the correlation was 100% compared with Illumina results, and that's true that with TMB it was over 90%, and the samples that have not a 100% of correlation, it was because of the threshold that we are using that is also impacted by the coverage. This is a little bit complicated to calculate the TMB, but the results were comparable with for the 3 biomarkers. Therefore, with the evaluation of these 3 parameters, for us was sure that the MGI platform was an alternative compared to Illumina sequencer, and also make that the implementation of more comprehensive, bigger panels are more accessible, since with the MGI technology we can obtain a better cost effectiveness during the sequencing. Well, after the validation of the process using the MGI chemistry and verifying the entire workflow in our facilities, we design different services that we are offering to our customers based on the OncoDEEP solution, as well as all the in-house expertise in pathology. Because we have large experience now in Eurofins. In pathology, we have a laboratory in Madrid that is only focused in this kind of services, and together with this knowledge in pathology and the OncoDEEP, we can offer the 5 different services that you have in the slide right now. As a result, we have able to offer OncoHub Tumor D, focused only in DNA analysis, that we analyze the 630 genes and also HRD, MSI, and TMB. Then the possibility to introduce the OncoHub Tumor DR, that includes both DNA and 22 genes analyzed by RNA. And additionally, with OncoHub Tumor Plus, it's possible to incorporate specific studies at protein level that assess response to immunotherapy, such PD-L1 and CD8. And finally, with OncoHub Tumor Max, a more comprehensive services is also available, included advanced protein level studies in accordance with international guidelines and scientific evidence for target chemotherapy and immunotherapy, as well as MGMT promoter methylation analysis. Of course, during the implementation, not everything was easy and nice. If the audience have experience in the implementation of new technologies in the laboratory, they know that not always is so easy as we would like it. But in order to have 100% operational and meeting the highest standards of quality and performance, we encountered a series of challenges. One of that was adapt workflows to existing services and ensure proper logistic quality in the collection, transport, and storage of the samples. This is quite different if you used to work only with germline to introduce this kind of type of samples in the clinical routine. Then working with tumor samples involves handling material that is much more sensitive and difficult to manage compared to germline studies. Therefore, it is essential to ensure that high sensitivity and specificity and have sufficient quantity and quality of DNA to avoid incomplete results of false negatives. We show in the data of the verification of the MGI technology that with 5-6 nanograms of star material of DNA, there is one variant client that it was not possible to identify that with 30 and 100 nanograms can see perfectly. Therefore, it is mandatory to obtain enough DNA to avoid these incomplete results of false negatives in the results. And also, we show certain difficulties in the RNA-based studies because the challenge during the extraction process due to sample degradation, which makes it essential to have a highly optimized purification protocol for the RNA to introduce into the procedure. The data analysis must also provide a fully automated and validated system, not only for variant detection, but also for genomic signatures, minimizing the need for manual interaction during the interpretation of the process. And lastly, and for me, as I said before, one of the most important points, no matter how simple the interpretation process may seem through advanced interpretation tools, it is absolutely essential to have a team of geneticians and clinicians who can analyze the results and generate clear, straightforward reports for oncologists, as well as be available at all times to support them with any questions or complete cases analysis. So, from my side, I'm finished. Thank you very much to all of you, and I give back the word to Linda.
Thank you, Alejandra, for sharing your experience with us. So now it's time to open the Q&A session. So as mentioned in the chat, you should be able to ask your questions through this channel, and all our speakers will be able to join the how to say the interaction. Will be able to interact with you during the Q&A session. So please, yes, do not hesitate. Share all your questions, and we will be more than happy to answer it. Maybe I can share also some questions we already received before the webinar from people who will not able to attend the webinar. So I received some questions for OncoDNA, some other for MGI. So maybe we can start with one question we received for OncoDNA. It's related to the quality data. Can we get access to sample metrics, coverage, conformity, etc., to identify quality issues? You are on mute, I think, Conrad.
My apologies. So indeed, there are two things which happen when working with OncoDNA. First of all, the team will verify the data quality before the analysis is launched. So there is already a check before we launch the analysis in our BioIT tools. And then, of course, later on into the system, into the OncoKDM system, there's a specific page where you can consult all the quality criteria, all the quality data for each sample. Also, you can download, as I mentioned, all the raw data, which will allow you to even look more into the details of the quality criteria and the quality for a specific sample.
Thank you, Conrad. Another question. So I should another one coming for MGI. How many samples can be sequenced on the G400 when using the OncoDEEP Kit?
So for G400, we can run around 96 samples, actually even a bit more, but then let's say what's fitting the kit setup is 96 reactions, so it's would be standard thing to do would be 96 samples per run per flow cell.
Great. Thank you, Philips. Maybe I can
If
continue.
I can say something extra to Philip's, and that is, it is possible also to run because Philip said they can be 96 samples simultaneously, but you could run more. And exists a possibility to run simultaneously samples from OncoDNA with other solutions, always if you have the same sequencing condition. That means if you are running exomes, hereditary cancer, and other solutions that they have parent 100 or parent 75, then you can run together and then make more cost effective the sequencing simultaneously on the MGI platform. Then you don't have this limitation according to the number of index, etc. Because if you are combining with other solutions, then you can make more cost effective the sequencing.
Yeah,
Sorry.
exactly. And one thing, yeah, that's a very good point. Thank you for raising it. And one thing to add, that we have four lanes, and basically each lane is separate, and we can combine it. We're limited in this situation exactly only by the length of sequencing.
Regarding variant detection, when editing a variant, does this change apply to all the other samples? I think it's for you,
I guess this is
Conrad, and also Alejandra, based on your experience.
so. As mentioned with OncoKDM, we have our OncoKDO database, which is maintained by our scientists. So we take the assumption that what our people define is the correct truth for OncoDNA. So the data as in OncoKDO will always be displayed for a patient as it is in OncoKDO. Of course, if a lab says I have another opinion for this specific case, there is the possibility to change the biological impact into the platform for that specific patient.
Thank you, Conrad. And I think I will finish maybe with the last question to Philips. So it's someone who know that MGI provide automation system and was wondering if MGI have a script designed to prepare the library from OncoDEEP.
So, as I was mentioning during the presentation, this is one of the projects that we're working on, and the vision is that we see SP100 automation fitting really well into this pipeline. So there most likely, I cannot say exactly when, but relatively soon there should be a solution on the automated platform. And of course, this is SP100, which is optimal for, let's say, around 16 samples per run. But for a higher throughput labs, we can also integrate a solution, have a script for SP960, which can run 96 samples in parallel.
Thank you, Philips. Maybe I can also share this one who looks very interesting regarding the quality data for OncoDNA. So the question is, how do we know if we are missing some targeted regions?
Well, also in the platform there is a file that you can download which will indicate the coverage of each region or the lack of coverage in certain regions. So there is the possibility exists to download a file which contains all of this information about the regions that were not properly covered in the sequencing.
Thank you, Conrad. So just for those who are interested, maybe I can share in the chat the link to download the application note. Yeah, here I think the link is available now. And I feel like our speakers have been really clear during their presentation and cover all the topics that our attendees needed. So I think we can close the webinar for today. Thank you again for all your great presentation and hope to see you soon in the future.
Thank you very much.
Thank
Thank you,
you.
everyone.
Bye-bye.
Bye.
Thank you. Goodbye.